Welcome back, everyone. Today we're diving deep into something pretty amazing, generating blood cells. And not just any blood cells, but ones created from human pluripotent stem cells.
Definitely cutting-edge science.
We've got this fantastic review article just chock full of the latest research.
A lot to unpack there.
Oh, absolutely. We'll break it down for you. Ever heard of using 3D mini environments to grow cells?
Or universal blood that could work for anyone.
That's just the tip of the iceberg, folks. Get ready.
Yeah, it's a field that's really exploding with possibilities.
So let's start with, the basics. Our bodies, they're constantly producing blood cells, right? It's called hematopoiesis, but it's way more complex than just, one process.
Nature actually uses 3 different waves to make those blood cells, each one a little more complex than the last.
So why is that final definitive wave? Why is that the holy grail for scientists?
Well, that's where we get to the really good stuff. The definitive wave is where we unlock the ability to create hematopoietic stem cells, or HSCs for short.
HSCs, okay.
Yeah, those are the master cells,
Okay.
source of all our blood cells throughout our lives. If we can replicate that in the lab,
Mm-hmm.
well, we could potentially have an unlimited supply of any blood cell type we need.
Wow.
Think about it. Treating blood cancers, immune deficiencies.
Oh, I see. So instead of relying on limited sources like bone marrow or cord blood,
Exactly.
we could just create a blood cell factory.
Right? In the lab.
Amazing. So what makes these human pluripotent stem cells, or hPSCs, what makes them the key to all of this?
Well, these cells are pretty special. They're like blank slates with the potential to become any cell in the body.
Wow.
We're talking embryonic stem cells, or ESCs, which come from embryos. And then there are induced pluripotent stem cells.
iPSCs.
Yep, you got it. iPSCs are adult cells that have been, reprogrammed back into that stem cell-like state.
So like taking a skin cell and turning it into a blood cell.
Basically.
That's mind-blowing.
It really is.
Okay, so what advantages do these hPSCs have over, the traditional sources?
It's a game changer on a few levels. First, availability. Think about it. Generating a huge supply of blood cells without needing donors.
Okay.
That's huge. Then there's personalized medicine. You could use a patient's own cells to create perfectly matched blood cells.
So no risk of rejection.
Exactly. No rejection, no side effects. And the ultimate goal, creating universal blood cells that could work for anyone, no matter their blood type.
No more donor matching issues. That's incredible. This could really, revolutionize transfusions and transplants.
It really could.
Okay, so let's get into the how. How do scientists actually turn these hPSCs into the specific blood cells we need?
It's a delicate dance mimicking the natural steps of hematopoiesis, but in a very controlled environment. Scientists have come up with a bunch of techniques over the years. They started with something called embryonic bodies, or EBs.
EBs. Those sound almost like letting the stem cells just do their thing, but in 3D.
You got it. These 3D clusters of cells, they're a good starting point, but you don't have much control over the types of cells they make. So scientists then moved on to co-culturing hPSCs with stromal cells.
Stromal cells?
Yeah, they're like support cells, creating a more natural environment for blood cell development.
Oh, I see. Like making a little ecosystem in the lab.
Exactly. And researchers have gotten really good at fine-tuning these methods, moving towards more controlled techniques, things like monolayer-based, serum-free differentiation. That gives them way more control over the environment.
Makes sense.
But the really exciting stuff is happening with 3D systems.
3D systems. Now that sounds like the cutting edge. Tell me more.
We're talking about environments that even more closely mimic the real-life conditions where blood cells develop. Think 3D self-assembling peptide hydrogels, microcarrier systems. They provide all the right cues for the cells to become specific blood cells.
Like tiny high-tech nurseries for these cells.
Aha, yeah, I like that. And then there's a really wild technique in vivo teratoma formation, using the body itself, like in immune-deficient mice, as a living incubator.
Okay, now that's getting really interesting. How does that work?
Well, scientists inject hPSCs into these mice,
Uh-huh.
and they form these benign tumors called teratomas.
Okay.
These teratomas actually contain a bunch of different tissues, including blood cells. And the amazing part is, scientists have been able to isolate functional HSCs from these teratomas.
Wow, it's like we skipped ahead a few chapters in this story.
Yeah.
We've got the basics of hematopoiesis, the potential of hPSCs, and all these fascinating techniques from EBs to 3D environments, and even using living organisms as incubators. This is amazing stuff.
It's just the beginning.
But I want to get into the specifics. What about creating those immune cells? Like T cells, NK cells, B cells?
Oh yeah, the immune cell generation, that's a whole other world of progress and challenges. Let's dive in. T cells, now those are fascinating. They're like the conductors of our immune system,
Yeah.
recognizing and destroying those infected cells, coordinating the whole immune response.
Keeping everything in check.
Exactly. But getting those freshman T cells, the ones that haven't encountered any invaders yet, generating those from hPSCs,
Yeah.
that's been a real challenge.
What makes creating those naive T cells so hard? I mean, it seems like we're getting pretty good at mimicking, natural processes in the lab.
Well, think about it. T cells normally mature in the thymus, right?
Right, that little boot camp for the immune system.
Exactly. And replicating that environment, that complex process in a dish, that's tough.
Yeah, I can imagine.
But scientists, they always find a way, right?
Right.
They've been using these co-culture systems with stromal clouds that express this protein, DLL1, which is like crucial for T cell development.
Okay.
But they're not stopping it. Now they're building 3D artificial thymic organoids.
Whoa, mini thymuses in the lab.
Pretty much. Mini thymus environments to try and get those T cells to mature properly.
So we're not just mimicking the process, we're recreating the whole training ground.
Exactly.
Wow. And is it working? Are these mini thymuses producing those naive T cells?
The results are looking good. The T cells from these 3D environments, they're showing a much more naive phenotype, much closer to what you'd see in a real thymus.
That's amazing. What about those antigen-specific T cells? The ones that can target, like, specific viruses or cancer cells?
Oh, yeah, that's where it gets really interesting. Imagine having a whole army of T cells trained to attack a specific target.
Like a personalized immune army?
Exactly. And scientists are figuring out how to do it.
How?
One way is they take mature T cells that already have that antigen specificity, and they kind of rewind them, turning them back into iPSCs. Then they differentiate them back into T cells, and boom, they retain that original targeting ability.
That's incredible. And is this how they're doing that CAR cell therapy? I've been hearing so much about that.
That's right. CAR cell therapy, it's revolutionizing cancer treatment, especially for certain blood cancers.
It sounds like sci-fi, honestly.
It kind of is. Imagine taking a patient's own T cells and you equip them with this thing called a chimeric antigen receptor, or CAR.
Okay.
And this CAR, it's like a GPS system.
A GPS for the immune system?
Yep, it guides those T cells straight to the cancer cells.
No wonder it's so effective.
And now scientists are trying to generate CAR T cells from iPSCs, which could make this therapy even more accessible.
Wow, this is really groundbreaking stuff. Okay, we've covered T cells. What about NK cells, those natural-born killers?
NK cells, they're like the rapid response team, always patrolling for trouble.
All right, always ready to go.
No need for prior sensitization or anything.
Yeah.
And what makes them so exciting for immunotherapy is their HLA-independent killing action.
HLA independent, meaning they don't have to be perfectly matched to the patient.
You got it. And that opens up the possibility of creating, off-the-shelf cell therapies.
That could work for anyone.
Exactly. And scientists have already come up with a few ways to generate NK cells from hPSCs.
And are they making CAR NK cells too, like they do with T cells?
Yeah, absolutely. Engineering those NK cells to target specific cancers.
So CAR cells and CAR NK cells, are they like basically the same thing?
Well, they both use this amazing genetic engineering to create those targeted immune responses.
Yeah.
But some researchers think CAR NK cells might have some advantages in certain cases.
Oh.
Yeah, potentially fewer side effects than CAR T cells.
Interesting. It seems like there's no stopping this immune cell engineering train.
It's a fast-moving field, that's for sure.
So much potential.
Absolutely.
Okay, so what about B cells? Those are the antibody factories, right?
Right. Essential for producing those antibodies that neutralize pathogens.
Yeah.
But they've been a little trickier to generate from hPSCs.
Oh, how come?
They need a more complex differentiation process, kind of like a multi-step recipe with really precise timing and ingredients.
So are we making any progress with B cells?
Oh yeah, definitely. We've seen some success in generating B cells that express IgM, which is a type of antibody,
Okay.
but creating the full range of specialized B cells, the ones that target specific pathogens, that's still a challenge.
Sounds like B cell research is a little behind compared to T cells and NK cells.
It's a tough nut to crack.
Well, we've talked a lot about the immune system, but what about those other crucial blood cells, the ones that carry oxygen and help us stop bleeding?
You mean red blood cells and platelets? Now that's where things get really exciting with clinical trials already underway.
Really? Wow. But red blood cells, platelets, I mean, they're like the essential workers of our circulatory system. It's pretty wild to think we're actually close to making them in the lab.
It really is a true testament to science and engineering.
For sure. Let's start with platelets. Those tiny guys are crucial for blood clotting,
Absolutely. They prevent us from bleeding out, even from the tiniest cuts. And the demand for platelet transfusions is huge, but, well, relying on donors has its downsides.
So how are scientists tackling the whole creating platelets from hPSCs thing?
They've got this three-step process. First, they start with those hematopoietic stem cells, the HSCs.
The master blood cell making.
Yeah, exactly. Then they nudge them to become megakaryocytes, which are basically the platelet precursors.
Okay.
And finally, they coax those megakaryocytes into releasing platelets.
Wow. So we're basically like recreating the entire platelet production line, but in the lab?
Precisely. And to, really boost efficiency, scientists are using all kinds of tricks.
Tricks.
Huh? Well, techniques, they're co-culturing with feeder cells, genetically modifying the cells, even using bioreactors to create like the perfect platelet-making environment.
Bioreactors, those sound pretty high-tech.
Oh, they are. Think of them as like high-tech chambers that mimic those physiological conditions, and that allows for, large-scale platelet production.
Makes sense. But wait, you mentioned universal platelets before. What's that all about?
Oh, right. That's where it gets really crazy. Scientists are working on making platelets from hPSCs that can be used for anyone.
No way. So they could work like regardless of blood type.
Exactly. They're removing the HLA genes. Those are the ones that cause the immune system to, freak out and reject the transplant.
Oh, right, right.
So no more matching issues, no more worries about rejection.
That's huge. That could totally change how we do transfusions. Okay, so what about red blood cells? Any progress on that front?
Oh, yeah, big time. In fact, in 2022, scientists did something incredible. They actually transfused lab-grown red blood cells into a human volunteer.
Whoa, hold on. Lab-grown blood transfusions. Is that even possible?
It is. It was a small trial, of course, but the results were promising. It really opens the door to a future where blood shortages could be a thing of the past.
That's amazing, but I'm sure it's not all smooth sailing, right? There must be some challenges.
Oh, no, always challenges. One of the big ones with red blood cell production is getting those precursor cells, the erythroblasts, to produce enough hemoglobin.
Hemoglobin, the stuff that carries oxygen?
Right. And then there's the whole process of getting rid of the nucleus in those cells, the enucleation process. It's not as efficient as we'd like it to be.
So there's room for improvement. Still, it's pretty incredible that we're already at the point of doing human trials with lab-grown blood. This whole field of, generating blood cells from hPSCs is just mind-blowing. But I'm sure there are bigger, broader challenges too, right? Things that researchers are still trying to figure out.
You're absolutely right. There are a few key challenges. We've got to be careful about tumorigenicity.
Tumorigenicity. That sounds scary.
It is a concern, for sure. It's the risk that these cells could, start growing uncontrollably. Then there's immunogenicity, the chance that the body will reject the transplanted cells, even if they're a perfect match. And we're also still working on overcoming heterogeneity.
Okay, a lot to unpack there. Let's start with tumorigenicity. How are scientists addressing that?
Well, there are several strategies. One is to use very sensitive cell sorting techniques
Okay,
to make absolutely sure that the final product only has the blood cells we want, no lingering stem cells that could potentially cause trouble.
makes sense. So it's like super careful quality control.
Exactly. Another approach is to engineer the hPSCs to give them like a suicide gene.
A suicide gene.
It's a safety mechanism, basically. If the cells start acting up, we can trigger it to eliminate them.
Oh, wow, that's pretty ingenious. What about immunogenicity? How do we stop the body from rejecting those lab-grown cells?
Well, this brings us back to those universal cells we talked about.
Ah, right, right.
By knocking out those HLA genes that trigger the immune response, we're basically creating cells that are like invisible to the immune system. They can be transplanted into anyone without the risk of rejection.
Invisible to the immune system. That's amazing.
It really is a game changer. And it's not just theoretical, it's already being used for platelets and K cells, even T cells.
Wow. So those universal cells could really be the key to making all of this work. Okay, what about that last challenge? Heterogeneity. What was that again?
Heterogeneity, yeah. That's just the fact that even with all these precise techniques, we still get some variation in the cells we produce. It's like baking cookies. Haha, yeah. You follow the recipe perfectly, but some cookies might come out a little crispier than others.
Ah, I see. So it's like inconsistent results. How do we fix that?
It's all about refining those recipes, those differentiation protocols. Scientists are always tweaking the mix of growth factors, signaling molecules, and culture conditions to make sure those cells mature and function exactly how we want them to.
So it's a constant process of improvement.
Absolutely. This field is constantly evolving, always pushing the boundaries.
Yeah, that's definitely clear from everything we've talked about. It's been an incredible journey exploring the world of hPSC-derived blood cells with you. I mean, this technology, it has the potential to revolutionize medicine.
I couldn't agree more.
Think about it. Blood cancers, immune deficiencies, genetic disorders, even traumatic injuries, all potentially treatable with these safe and effective cell therapies. It's mind-blowing.
And it's all thanks to the tireless work of so many dedicated scientists.
Absolutely. And for our listeners out there, think about this. What if one day we could all have our own personalized blood bank, created from our own iPSCs, a ready supply of perfectly matched cells for anything, whenever we need them? It sounds like sci-fi, but the future is closer than we think.
It's an exciting time to be alive.
It really is. Thanks for joining us on this deep dive, everyone. And until next time, keep exploring. 1718 8s.